So in the previous class, we have discussed about the detail of the testis now. So after this segment, this was the testis. Now the next part is the epididymis. So this is the epididymis. Okay. So the epididymis, what does this means? Epididymis, that is epididymis. So if we divide this thing, so the word will be epi and didymis. So epi means, you know that epi means above, and didymis means testis. So epididymis is a structure that lies above the testis. You can see here. So this is the epididymis, and it lies above the testis. So this structure is epididymis. So it is a comma-shaped organ. You can see here. This is an almost comma-shaped organ, and it lies on the posterior border of the testis. So first of all, the lining epithelium. So the epididymis is lined by pseudostratified columnar epithelium and with the underlying layer of smooth muscle. So how epididymis is formed? So you can see here the seminiferous tubules. All the seminiferous tubules converge here on the retitestis, and they form a network that is called the retitestis. And from this retitestil, some ductules arises, and these ductules are called efferent ductules that ultimately fuse to form the head of the epididymis. Then head of the epididymis descend downwards and form the body of the epididymis, then the tail of the epididymis, and the tail of the epididymis ascends and continues as vas deferens. Okay. Now, if we see the histological structure, these epididymis, it is a coiled structure. So on a cross section, the structure will almost looks like the structure of testis, but there is a difference. So what is the difference here? Yes. So on the slide of testis, we can see here there are different lobule. Okay, so in testis there was different lobule, but there is no such lobule in the seminiferous, in the epididymis. And there was tunica albuginea, a thick connective tissue layer. This layer will be also absent in the seminiferous tubule. So we will see only this underlying, so this basement membrane, above which there will be, if we zoom one lumen, so there are the pseudostratified columnar epithelium, and on the free surface you can see here some small thread-like structure, long thread-like structure. So these are the sterocilia. Okay? So pseudostratified columnar epithelium will be present in the seminiferous tubule with some sterocilia. So they actually increase the surface area for the reabsorption of degenerated sperm. So some sperm, when they will come to the sperm will go to the epididymis, some sperm will be degenerated and they will be reabsorbed by the epididymis. So what are the functions of epididymis? function.
Maturation of spermatozoa.
Yes, one important function is sperm maturation and storage. And another function is to diperidamys also helps the sperm to propel into the vas deferens by the peristaltic contraction. So peristaltic contraction of the smooth muscle. Okay? So these are the overall function of the epididymis. Now, one important structure, next important structure is vas deferens. So to this segment, this was the head of the epididymis, body of the epididymis, tail of the epididymis. Then the next structure will be the ductus deferens or vas deferens. So it is actually a continuation of the tail of the epididymis, and after formation, it enters into the abdomen as a component of the spermatic cord. You know that the spermatic cord passes through the inguinal canal. So spermatic cord containing the vas deferens that will enter through the inguinal canal into the abdominal cavity. This vas deferens, it will also lined by pseudostratified columnar epithelium, almost like the epididymis. So here, so this is the testis. After formation, these are the ductus deferens. So these ductus deferens, it will undergoes the inguinal canal here. This is the deep ring. This is the superficial ring. So through the inguinal canal, it will enters into the abdominal cavity. Then, so it is the posterior surface. So after, so it will come over the urinary bladder, and ultimately it will form a dilated part. This dilated part is called the ampulla of the vas deferens. And there is another accessory gland here, that is seminal vesicle. So these vas deferens will unite with the duct of the seminal vesicle inside the prostate, that is another accessory gland of the male reproductive system. And ultimately, secretion from the vas deferens, secretion from the seminal vesicle, and some secretion from the prostate, that will comes out during the ejaculation through this urethra. Okay, so this is the overall pathway of the sperm. So if we starts from here, so this question is usually asked in the oral examination. So pathway of the sperm, at first from the seminiferous tubule, then from here, then from the retitestis, from retitestis to head of the epididymis, then body, then tail, then vas deferens, then into the vas deferens will here, then ampulla of vas deferens unite with the seminal vesicle into the prostatic part, then through the urethra into the female reproductive system. So this is the overall pathway of the sperm. Now seminal the seminal vesicle. So this was the seminal vesicle. So seminal vesicle is the accessory gland, so it will secrete some substance. So the function of the seminal vesicle is to it secretes it will secrete some alkaline fluid that will neutralize the acidic environment in the female reproductive system, and it will provide fructose. One important function that it will provide So it is at first is alkaline secretion that it will contain fructose. So fructose is a type of glucose. So it will provide the ATP or the nutrition for the sperm that will be used for the motility and viability. Then the prostate. So the secretion of the prostate, it is called prostatic fluid. And another important thing that there are some glands in this region that are the bulbourethral gland. They will also secrete some alkaline fluid to neutralize the acidic environment of the urethra, okay? And some lubricating secretion. So, and this is the part of male urethra. I think you have learned about this well, so I'm not going in detail about this thing. So now one important question, that is the pathway of sperm. It is very important, so you will learn in detail. I'm telling you again. So at first seminiferous tubule, then retitestis, then efferent ductule, head of the epididymis, body of the epididymis, tail of the epididymis, then vas deferens, and vas deferens unites with the seminal vesicle inside the prostate. So along with the secretion of the seminal vesicle, vas deferens, and bulbourethral gland, the sperm will goes through the urethra to the female genital tract. So this is overall discussion of the male reproductive system. So the most important topic of this class is the structure, histological structure of testis, important. Then most important, blood supply of testis, then content of spermatic cord, that is important for oral, speak, and written also. Then another important question from here is pathway of sperm. Another important is blood testis barrier. Another important is short note on Sertoli cell. And another important is that is related to embryology, but from this subject, that is the spermatogenesis, spermiogenesis. So that will be included in the embryology, but from this topic. This is all about the male reproductive system. Today we will discuss about another class, that is renal system or urinary system, and on the next class we will discuss about the female reproductive system. Any question from the male reproductive system? Anyone?
Sir, epididymiসের যে হিস্টোলজিক্যাল স্ট্রাকচার, ওটার মধ্যে লুমেন দেখি না কেন?
সরি, ওটাতে কি দেখি না?
লুমেন স্যার, আপনি যে বললেন যে টেস্টিসের
না আমি লুমেন মিন করি নাই। আমরা এখানে যেরকম টেস্টিসের ক্ষেত্রে যেরকম আমাদের লোবিউল দেখা যায়, ওকে, এই যে কানেক্টিভ টিস্যু সেপ্টা দিয়ে যে লোবিউলের মতো ডিভাইড ছিল, মুছে দেখাই আমি। So here এই যে দেখেন, এখানে যেরকম কিছু লুমেন ছিল, কানেক্টিভ টিস্যু সেপ্টা ছিল এরকম, these are the connective tissue septa. So epididymis এই connective tissue সেটটা থাকবে না। And
Yes,
এখানে
sir.
on the outer border এখানে একটা thick connective tissue টিউনিক অ্যালবুজিনিয়াল লাইনিং আছে, কিন্তু এটা আবার এখানে যেমন আমাদের এপিডিডাইমিসের ক্ষেত্রে there is no such septa or lobule. একটা জিনিস হচ্ছে টেস্টিসের ম্যাগনিফাই there are different multiple layers of cell. Okay, one layer, two layer, three layer, 4, 5. So multiple layers of cell due to the different stages of the spermatogenic cell. But in case of epididymis, only single layer of cell you can see here, almost pseudostratified, but tall columnar cell can be identified. Okay, mostly long columnar cell. Okay. And there are some stereocilia. In case of testis, there is no stereocilia. The cells are mostly rounded, and numerous sperm can be seen in the lumen. Okay.
Yes, sir. Thank you,
And
sir.
in the testis, in between the lumen, there will be interstitial cells of the lady. But in case of seminiferous tubule, there is only connective tissue. In between the lumen, there is no definite cells. So now
Yes, sir.
you will have a break for 5 minutes. If anyone have any question, you can ask. After 5 minutes, we will start the urinary system. Anyone have any question? Any question? But প্লিজ আপনারা একটু পড়াশোনা করবেন। আমাদের পড়াশোনার অবস্থা খুব খারাপ, একটু পড়াশোনা করবেন, নাহলে খুব খারাপ হয়ে যাচ্ছে। আমাদের যে অবস্থা, আমি এটা এরকম এক্সপেক্ট করিনি, সত্যি কথা।
স্যার,
ইয়েস
স্যার, প্রস্টেট কার্সিনোমাতে যদি প্রস্টেট রিমুভ করে না হয় তবে কি পার্সনের রিপ্রোডাক্টিভ কোনো ডিফেক্ট দেখা দেবে?
নট লাইকলি। অ্যান্ড প্রোস্টেটিক কার্সিনোমাতে যদি রিমুভ করে খুব একটা ডিফেক্ট হবে না আর কি, কারণ seminal fluid শুধু তো prostatic, not only the components of the semen is not only the prostatic fluid, but mostly seminal fluids from the seminal vesicle, testis, etc. And testicular carcinoma is very unlikely in early age. It is mostly okay. And the prostatic carcinoma are mainly mostly benign type prostatic hypertrophy. So in that case, mostly the hypertrophic tissues are removed. Whole testes, prostate are usually not removed. সবাই আছেন আবার? হ্যালো।
Yes, sir.
Yes, sir.
Okay. So I think you have completed your urinary system both in your lecture class as well as in your tutor class. So I will go through this topic very rapidly, and I will try to cover the histological aspect. So if anyone have any question, please ask during the class. And if you okay. So today's topic is urinary system. So first of all, what are the components of urinary system? So there will be 2 kidneys, 2 ureter, 1 for each kidney, and single urinary bladder and a urethra. So there's 2 kidney, right and left kidney, right and left ureter. Then 2 ureter will converge into a single urinary bladder, and the urinal will ultimately will be voided through the urethra. So from kidney, whenever in your oral exam or when you will get your viscera there. So one thing you will have to remember that the kidney, the both kidney are not lies at same level. Why?
So due to
Please
the
answer.
presence of the liver
Presence of the liver.
Right
on
lobe of
the right
liver,
side.
kidney side says the below than left.
Another one. Another important is how you will decide which one is right kidney and which one is left kidney, because in your college the viscera is both the kidneys with both these vessels are and the ureter are together. So how will you identify which one is right and left kidney? Then answer is you will have to identify which one is vena cava and which one is aorta. So I think whenever you had physical class there, so aorta or artery was thicker wall, vena cava or vein has thinner wall. So by this thing, you will at first identify the vena cava and aorta. So the vena cava side will be the right kidney and the aorta side will be the left kidney. So from here, there is one important thing you will be asked in your oral, that right renal vein or left renal vein, which one is longer? Common sense. As
The
inferior vena cava is on the right side, so which vein will be longer?
left
Left renal vein.
renal.
Left renal vein.
Opposite thing in case of the aorta. Okay. Another important thing you will be asked that left renal vein, it also receives some tributaries. For example, right renal vein directly is drained into the inferior vena cava, but left renal vein, it receives some other vein. For example, one important is this one, gonadal vein. Clear? So these are the basic questions you will be asked. Then the function of kidney, I think you're familiar with this thing. So first, regulation of the balance between water and electrolyte. So water balance and electrolyte balance, and it also help take part in the acid-base balance. Then excretion of metabolic waste product along with excess water and electrolyte, and excretion of many other bioactive substances, some drugs, and it also helps in regulation of arterial blood pressure by secretion of renin. So kidney is a very essential organ. So our body has two kidney, but an individual can survive with one kidney also. Okay. So very important and crucial functions are performed by kidney: acid-base balance, electrolyte balance, water balance, and also maintenance of the arterial blood pressure. Kidney function কোনো ভুল না হয়, okay? So before going to the figure, so the kidney has outer cortex and inner darker medulla. The outer cortex that will contain some corpuscle and some tubules, and inner medulla will contain some straight tubules. I'm showing you the figure here. So cortex will be the outer reddish brown part. So outer this part. So this is the renal cortex, and this part, this is the medulla. So outer cortex. So this is cortex, this is medulla. So in the medulla, there are some triangular structure here. So they are called renal. What is this renal?
Renal pyramid.
You have class renal pyramid. So how will you divide kidney into renal lobe? Bolen.
So one renal pyramid and a cortical arch will make a renal lobe.
Not only the cortical arch, because interlobar যে part, So এই পার্টের তো হাফ আসবে এখানে। So,
Half of
these
each.
are the
Yes,
renal
half of
pyramid.
the yes.
These are the cortical arch. So, and these are the
renal cortex.
these are the renal column. Okay. So this gap is renal column. So renal pyramid with its cortical arch and the half of the uh, renal column on each side of the pyramid, that will form the renal lobe. Okay. And the gap in the this empty space in the medulla, that is called the renal sinus. So this renal pyramid, they will contain numerous uh, tubules that will ultimately converge to form the minor calyx. So these are the minor calyx. So multiple minor calyx. So renal, you have a kidney model in your college. So that model is large model, almost similar to this figure, this diagram. So you will have to identify. So this renal pyramid, renal cortex, cortical arch, sinus, renal column, major calyx, minor calyx. So you will have to get a clear conception about this figure, so that whenever you will get those that uh, diagram on the model, so you will be asked about this thing. So please concentrate. So renal, this is cortex, this is medulla. So in medulla there will be renal pyramid and renal column. So what is renal lobe? This renal. So renal lobe will be this pyramid along with the part of renal column on each side and cortical arch. So it will combinedly form the renal lobe. And এই ছবিটা একটু ক্লিয়ার, আরেকবার বলতেছি আমি। So this segment, this is renal cortex. This segment, renal medulla. This pyramidal structure, that is renal pyramid, and in between these two pyramid is space that is called renal column. So are you clear about the renal cortex, renal medulla, column, and pyramid?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. And this junction, that will be the cortico-medullary junction. And now renal lobe. So this renal pyramid, along with the cortical arch here, this part, and part of renal column on each side of the pyramid, it will combinedly form the renal lobe. The pyramid, the apex of the pyramid lies at the collecting system that is called renal papilla, that will form the minor calyx here. Multiple minor calyx will combinedly form the major calyx, and the major calyx unites to form the renal pelvis. So these are the minor calyx, the larger major calyx, renal pelvis, and renal pelvis will continue as a ureter. Clear everyone, this figure?
Yes, sir.
Yes, sir.
Another important thing is mode of blood supply of the kidney. So it is very, very important for written. So I am not going in detail about this diagram. Mode of blood supply of kidney. Anyone? Can anyone explain?
Sir, renal artery are the branch of the lateral branches of the abdominal aorta.
Okay, renal artery, then.
Hilum দিয়ে ঢুকলো।
Okay.
তারপর স্যার lobar artery.
Lower artery আগে আরেকটা segment আছে. Another segment before lower artery.
Arcuate arteries.
Arcuate artery is this one. Arcuate. These are the
Segmental arteries.
yes, segmental artery, then lobar artery,
Yes, sir.
then
Arcuate arteries. No.
Interlobar artery.
again mode of blood supply of kidney very important from renal artery to renal vein. This total circuit you will have to draw and label some in some case, or you will be asked in the oral or written. Okay, this is very important. So now composition of the renal cortex. So the cortex will consist of renal corpuscle along with the convoluted tubules and straight tubules of the nephron, and some collecting tubules, collecting duct, and vascular supply, where the medulla will be contain the straight tubule, collecting duct, and the capillary network. So this term is very complicated. At first, we will see some figure of the nephron. Then we will come again to this slide to see what are the composition of renal cortex and renal medulla. So nephron. We know what is nephron. What is nephron?
Structural and functional unit of kidney.
Structure and functional unit of kidney.
The structural and functional structure of the kidney. So renal, this is nephron. Okay. So total this thing, this from here, this thing, this thing, this total thing actually combines to combinedly called nephron. So the nephron consists of two parts. One is renal corpuscle and renal tubule. So renal corpuscle is this circular or oval mass. So it will contain some glomerulus. So this glomerulus is actually a tuft of capillaries, okay, and some a capsular structure within which this glomerulus is placed. So this glomerular capsule, it has two layers. One is visceral layer, another is parietal layer. So the layer that actually encircles this glomerular capillary, that is the visceral layer, and the outer layer, this is the parietal layer. So one of the main function of the kidney is to filter the blood and production of urine. So arterial blood will come here and they will be filtered to form a filtration. So this glomerular renal corpuscle, that will be the first filtration station here. Then the renal tubules. So renal tubules are it's a long tubules and it contains different parts. In the proximal part, there is a convoluted part. So this is called proximal convoluted tubule. Then, and in the distal part, there is another convoluted tubule that is called distal convoluted tubule. Then the loop of Henle. So loop of Henle means there will be a loop like this. But for a loop, there will be a descending part, there will be a ascending part, and the loop. So the loop of Henle, it contains some straight tubule, proximal straight tubule, then the thin descending limb here, then the loop of Henle, then the thin ascending limb, and thick ascending limb. Okay. So the proximal convoluted tubule here, then the loop of Henle, descending tubule is divided into two parts. One is thick part, thin part. Ascending is again thin and thick. And there is a tube. Then in the distally, there is another convoluted segment that is called distal convoluted tubules here. Then the collecting tubules and multiple collecting tubules will drain into the collecting duct. So this collecting duct, numerous collecting duct will be placed here, the medulla, that will ultimately drain the urine into the renal sinus. Now here. So here the cortex consists of renal corpuscle along with convoluted tubules. That means in the cortex there will be renal corpuscle part of the nephron and the convoluted tubule. Both the proximal and distal convoluted tubule will be in the cortex. And the straight tubules of the nephron and part of collecting tubule and collecting ducts will be also present in the cortex. If we see here, if we magnify this part, so this is renal cortex. So you can see here the renal corpuscle, proximal convoluted tubules, distal convoluted tubules. So all the corpuscular part, proximal and distal convoluted tubule are present in the renal cortex, and as well as some part of collecting tubules and collecting duct. Part of collecting tubules and collecting duct will be also present in the renal cortex. So in the medullary part, the medulla of the kidney, that will mainly contains the mostly the collecting tubules, the, some loop of Henleys, okay, collecting duct. So these are the basic difference in the composition of the cortex and medulla. So in the medulla, there was a straight tubules, collecting duct, and some capillary network. So the nephron is the structural and functional unit of the kidney. Each kidney contains up to 2 millions of nephron. So nephron are the product responsible for the production of urine. It corresponds to the other secretory part of the other gland. The renal corpuscle I have mentioned already. It is a tuft of capillary. It is called glomerulus, and it is surrounded by a double layer epithelial cell that is called Bowman's capsule. And each capsule has one vascular pole. That is, for example, these are the tuft of capillaries. So if this is Bowman's capsule, it has two poles. One is vascular pole. So this is vascular pole, and from here, where the proximal convoluted tubule arises. So this is the tubular pole. So if you see the figure here, this is the vascular pole. This is the tubular pole of the glomerular capillary plexus or renal corpuscle. Now, so this is afferent arteriole. So this is afferent arteriole. Okay. So blood enters through this part. This is the glomerular capillary plexus, and then this is efferent arteriole. So you can see here the inner part of the glomerular capillary plexus that is lined by a specialized cell that contains multiple process here. So these cells are called podocyte. And you can see here there is a much gap in between these projections. So the capillary bloods are filtered through this pore, and the filtration goes through these proximal convoluted tubules. So this is the vascular pole, and this is the tubular pole. So another important thing that you should learn from the kidney, that is juxtaglomerular apparatus. So what is juxtaglomerular apparatus? It is a very common short note that comes in both MCQ, written, oral. So what is juxtaglomerular apparatus? It will contain, it is a composition of three types of cell. That is, one is juxtaglomerular cell, macula densa, and Lessig cell, and another type of cell. So these macula densa, I'm just telling here. So juxtaglomerular cell, you see here, is a modified cell of the, this is what? This is afferent arteriole, right?
Yes, sir.
So juxtaglomerular cell, these are some modified cell of the afferent arteriole. And macula densa, this is the distal convoluted tubule. So this cell, the cells of the distal convoluted tubule, along with the lies along the juxtaglomerular cell, that is called macula densa, and there will be interstitial cells here that are called Lessig cell. Those three types of cell, they will form the juxtaglomerular apparatus. We'll see their function later. So in the visceral layer of the capsule consists of highly modified branching epithelial cell, podocyte. I have shown the previous slide. So in the inner side, they are the podocyte, but in the outer layer of the outer side of the glomerular capsule, it is simple squamous cell. So the visceral layer, there will be podocyte, and in the parietal layer, that will be simple squamous epithelium. So on histological slide, juxtaglomerular apparatus looks like this. These are the tuft of capillary plexus. You can see there is a single flattened nucleus, that is the parietal layer, and the gap in between these two layers, this is called capsular space. Here, this capsular space is this one. So this is the electron microscopic figure of the podocyte. So this is one podocyte, and these are their pedicles. So this is the cell body, and these are the pedicles. And you can see here, you know that there are different in the cardiovascular system. We have learned about there are three types of capillaries: fenestrated capillaries, continuous capillaries, and sinusoid. So these are the fenestrated capillaries. You can see there are numerous pores in the capillary wall. And through this filtration slit, the filtration slit are this structure, the gap between pedicle of adjacent so these are the podocytes. So the filtration slits are the gap between the pedicle of adjacent podocyte. So now the histological difference between the different part of the nephron. First of all, proximal convoluted tubule, then loop-friendly distal convoluted tubule. But in your histological slide, how the first question is that how will you identify or differentiate between the proximal convoluted tubule and distal convoluted tubule? We'll come here later on this slide. So this is the important figure that I was talking about, the mode of blood supply of the kidney. So first of all, renal artery, then the segmental artery, then interlobar artery, that means the in between two lobes. So these are the interlobar artery. So these are the interlobar artery. Then arcuate artery means a arc like, this is a structure arc. So this arc like artery, these are the arcuate artery. So then they will form the interlobular artery. Now these interlobular artery will form the afferent arteriole, that will be glomerular in the glomerulus, that will form the glomerulus, and then they will form efferent arterioles. Now one important thing is that these efferent arteriole, they will form two capillary plexus. One is peritubular capillary plexus and vasorectal. Peritubular means capillary plexus around the proximal and distal convoluted tubular plexus, and they will form vasa recta. Vasa recta means vessel, recta means straight. So they will form these two straight vessel with intercommunicating channel. So these vasa recta and the different arteriole, they actually play a role in the different secretion or filtration or reabsorption of the part of the glomerular filtration. So from this vasa recta, then there will be reverse thing, interlobular vein, arcuate vein, interlobar vein, and renal vein. So this figure is very important. So in the portal system or kidney portal system, where is these two ports are sets of capillary? One is here, the glomerular capillary plexus. Another one is peritubular capillary plexus and vasa recta. Now this is the histological slide of kidney. So you will get this view on your microscope. But how will you identify this is kidney? Because you can see here there is a different multiple these are the renal corpuscle, numerous renal corpuscle. So renal corpuscle are mainly confined to the cortex, and in the medulla you will see only the tubules. So if you magnify here in the cortex, you will see only this thing. So these are the renal corpuscle, and this small gap, these are the capsular space, and in between this thing there are some tubular structure here. They are called medullary rays. This tubular structure in the cortex, they will be called medullary rays. They are actually the collecting duct and collecting tubules. Oh, sorry. Now these are the proximal and distal convoluted tubules here. So now, how will you differentiate between the proximal convoluted tubule and distal convoluted tubule? Because both are present here in this region, in the cortex. So how will you identify or differentiate between this proximal and distal convoluted tubule?
So there is no brush border in the distal convoluted tubule.
But in microscopic slide, to identify the brush border in this very smaller lumen is very tough. So we will see the slide whenever we get to the college. But now, in the proximal convoluted tubule, the filtration is much higher than the distal convoluted tubule. So the proximal convoluted tubule, there are numerous tall microvilli. Okay, and in the distal convoluted tubule, the microvilli are very short and small. So due to the presence of this tall microvilli, For example, in the lumen, and this distal convoluted tubule. So what is the difference you can see now?
Microvilli is longer in the proximal
In the
convoluted.
proximal convoluted tubule, due to presence of tall microvilli on histological slide, the lumen is almost non-recognizable. Okay, due to the presence of microvilli. So in the histological slide, we'll see the cell here, but the lumen is almost invisible due to the presence of these cannot be identified. We will see some this structure in the luminal part, but the distal convoluted tubule, the lumen will be clearly identified like this. So this is one tubule, this is another tubule, this is another tubule, this is another. So you can see here, you can see the cell here, but the lumen is supposed to be here, but due to the presence of tall microvilli of this cell, the lumen is not identifiable. Same goes to here. You can see here the cell, but the lumen is almost obliterated by these microvilli. So the tubule that lumen is almost unrecognizable. That is proximal convoluted tubule. Proximal convoluted tubule. You can see here, all this lumen is also not that much clear. So this is also proximal convoluted tubule. But the clear lumen here, you can see cell visible, but the lumen is clear. That is distal convoluted tubule. This is distal convoluted tubule. So if you see here, it is again proximal convoluted tubule. This is distal convoluted tubule. This one. So clear, everyone? What is the difference between proximal convoluted tubule and distal convoluted tubule in histological slide?
Yes, sir.
Yes, sir.
Now, this table is probably from your Zanpura. This is very important. So proximal convoluted tubule, there will be simple cuboidal epithelium. Distal convoluted tubule, simple cuboidal epithelium. But that is long microvilli, lumen is occluded. But here, the lumen is empty. In loop of Henle, both are lined by simple squamous epithelium, but tall ascending limb, that is thin limb by simple squamous, but the thick limb is cuboidal epithelium. Okay. Then the collecting tubule, there will be two types of cell, principal cell, intercalary cell, not that much important. Now, location is important. Proximal and distal convoluted tubule in the cortex. But, so the major functions, different secretion, you will just go through this table from your textbook. Very important major function, both in anatomy and most important for your physiology and biochemistry in the formation of urine. Very, very important. You have completed this renal system in your physiology or biochemistry.
Yes, sir.
Yes.
I think you know this thing better. So medullary. Now ureter. So what is ureter? So it is a muscular tube that conveys urine from kidney to the bladder by the contraction of thick smooth muscle layer in its wall. So now important, mucosa of the ureter, that is lined by transitional epithelium. Very, very important thing. Whenever you will get a transitional epithelium, then it will be asked, what is transitional epithelium and from where it starts? So transitional epithelium, so this is transitional epithelium. We have we have learned in detail about this in the epithelial tissue classes. So in a transitional epithelium, there are 5 to 6 layer of cell, but the apical cells are dome shaped and there is tight junction between the cells. And due to this tight junction, this epithelial cell doesn't allow any substance to go through it. So as we know that urine is urine contains different types of toxic substance. So wherever there will be urine, there will be transitional epithelium to prevent the reabsorption of harmful chemical substances. So from these minor calyx, from where? From minor calyx, transitional epithelium starts, okay? And it ends up to which part of the urethra? In case of female, throughout the whole urethra, there will be transitional epithelium, but in case of male, at the last part, there will be change of epithelium, but mostly that is lined by transitional epithelium. Okay. So the muscular layer of the ureter contains two layers of smooth muscle, so inner longitudinal and middle circular layer. The third longitudinal layer is added just near the urinary bladder, not throughout the ureter. So the lumen of the ureter will be looks like this, folded, very much folded due to the contraction of the smooth muscle. You can see here two layers of smooth muscle, one circular layer, and the outer side and the inner side there will be longitudinal muscular, and these are the transitional epithelium. Here 5 to 6 cell layer thick transitional epithelium is here, circular muscle layer and muscle layer. So the urinary bladder, so it is a thick muscular wall. Lining epithelium is transitional epithelium. Sorry. So it consists of outer and inner longitudinal muscle layer and middle circular layer. But in case of এদিকে যাচ্ছি না in detail, এটা আগে আমি আপনাদের হিস্টোলজিক্যাল ক্লাসে ডিসক্রাইব করছিলাম, সেটা হচ্ছে in case of urinary bladder during the resting condition or during not in extended, so empty bladder exhibits some mucosal fold that disappears during the distension of the bladder. So transitional epithelium is thicker than the ureter here. This is very important. The transitional epithelium begins in the minor calyx as two cell layer thickness, but gradually when throughout the course, the lining epithelium will add more and more cells. So this is the wall of the urinary bladder. This is also folded, but the muscle layer are not that much clear distinction because the middle circular layer is very thin, but the outer and inner longitudinal muscle layer is thicker. Now urethra যাচ্ছি না ডিটেল। সো ইউরেথ্রাটা আপনারা একটু দেখে নিন মেল এন্ড ফিমেলের মধ্যে difference and the different type of lining epithelium in case of different parts of the male urethra, posterior part, then membranous part, and the spongy part. Okay, so male and female urethra difference they can even. So any question up to this segment?
স্যার হিস্টোলজিক্যাল স্লাইড অফ দ্য কিডনি আবার রিপিট করা যাবে স্যার? শুনতে পারিনি তখন।
কোনটা
স্যার কিডনির যে হিস্টোলজিক্যাল একটা স্লাইড দেখালেন।
আচ্ছা দেখেন কিডনির যখন আপনার হিস্টোলজিক্যাল স্ট্রাকচার দেখবেন, সো ওভারঅল যখন 4x জুমে দেখবেন তখন অনেকটা এরকম দেখতে পাবেন। সো ইউ উইল সি মাল্টিপল this small round structure like this, and almost all the small round structure will be in the upper part. So up to this part where there will be this tubular structure, that is renal cortex, and where there will be this long tubular structure like this, that will be renal medulla. So in the cortical part there will be also some this long tubular structure like this here. So these are called medullary ray. So where there will be this longitudinal structure like this, that will be due to the collecting duct, collecting tubules. And this circular structure here, there is also some luminal structure here like this, but it's not visible in this figure. So that will be renal corpuscle and the renal tubule. So how will you differentiate between the corpuscle and tubule? Because the corpuscle will have a light gap in between these two layers. So this gap is called capsular space, and this is the glomerular capillary plexus. And these are the medullary ray, collecting duct and collecting tubules. So now then the second question is, how will you differentiate between the proximal convoluted tubule and distal convoluted tubule? Then so this is a tubular structure, this is another, this is another. So as the proximal convoluted tubule cell contains long microvilli, so the lumen will be obliterated and cannot be seen in the due to the presence of microvilli. Here, this is also proximal convoluted tubule. All these are the proximal convoluted tubule. But in the distal convoluted tubule, due to presence of very short or very few microvilli, the lumen are empty. So these are distal convoluted tubule.